Studying how this jellyfish is bioluminescent and makes its own light led to one of my favorite all-time scientific discoveries. It's got a protein called green fluorescent protein which absorbs blue light and emits green light. And if you take those jellyfish GFP genes and insert them into the genes of other organisms, you can make them do all sorts of cool stuff. So in 1962, Japanese biochemist and marine biologist Osamu Shimomura isolated that protein and figured out how the jellyfish fluoresces. And then 26 years later, in 1988, American molecular biologist Martin Chalfie heard about green fluorescent proteins at a seminar. And he went on to use it in his research subject, the transparent roundworm called C. elegans. He even got the cover of the February 1994 issue of Science for his paper on the subject. Now, one problem with studying proteins is it's pretty dang difficult to study specific proteins in living organisms and know which genes activate them at any given time. But you can throw some GFP genes next to the specific code for the protein you want to study. And then if you shine a black light on the thing, you can tell if your protein is being produced. Because if that protein is being produced, then the GFP is being produced too, and it fluoresces green. And that allows you to tag proteins and use GFP as a tracer. So you can see where proteins are inside of a cell and when they're being produced within the life cycle of the cell. And you can do things like bind it to cancer cells and get glowing, easy to find cancer. So then in 1995, American biochemist Roger Cien drastically improved GFP by slightly modifying the genes. And that made the fluorescent proteins brighter and developed a wide range of colors that would fluoresce. Like this little painting here was painted with genetically engineered fluorescent bacteria in a petri dish. With all those different colors of GFP, you can tag and study hundreds of different proteins at the same time. These images are called brainbows, and they're images of fluorescing brains, so you can see the intricate neural circuitries in them. In 2008, the three were awarded a Nobel Prize for their work on GFP. Thanks for watching, and if you can or could help me and my community support this GoFundMe to help save Amir and his family, I'd appreciate it, and they would too. Thanks to everyone who's helped at all so far, and huge shout out to whoever it was who just donated €1,000. Like, damn, thank you.